this is the right link, correct?
Yes, ma'am.
Okay, Amma, thank you very much. वॉल्यूम नहीं है। अच्छा, हाँ, ठीक है, इट्स ऑफ। Afrin, did you put in all groups, especially PhD, PG today to join?
Yes, ma'am. I'll just post it, ma'am.
Post it and ask class teachers.
Yes, ma'am. Yes, ma'am.
Hello, Dr. Subhashish. Good morning. Hello, Dr. Subhashish. Are you there? Lokesh sir, do you want to confirm with Subhashish sir if he could join?
Good morning, madam. I'm here.
Oh, okay, wonderful. Yeah, I was not sure whether you are there or not. Good morning, sir.
Good morning. Good morning.
Good morning.
I hope you can see me.
Can't see you.
I'm on screen.
Oh, yeah, I can see you now. Yeah. Perfect. All right. So, Team BNCP, how are we moving ahead?
Unmute camera on.
Yes, ma'am. We can start.
Camera on.
Okay, are we expecting few more participation? Do you want to wait for 2 minutes, Dr. Subhashish?
Yes, ma'am. We can wait.
I'm okay.
Okay, I see people are joining.
Good morning, everyone. Rajesh Parapi.
Hello, good morning, Dr. Rajesh.
How are you all?
Good. We had yesterday wonderful session.
Yeah, I was there for first half an hour.
Oh, okay. Wonderful.
Good
Great.
morning, Dr. Rajesh.
Good morning, Subhashish. How are you?
All good. How about you?
All well. Thank you.
Yeah. So today we'll have a PhD and innovation. Very much looking forward to listen to those entries, especially solid orals, both rather all. So just run the video for a few minutes, and then in 3 minutes we will start. When Lokesh sir and Sujata madam gives us heads up. Okay.
Yes, ma'am.
Okay. Dr. Parizad, join.
Yes, good morning, everyone.
Oh, wonderful. Good morning. Always ACG team on time.
Yeah, before time.
Before time, correct? We cannot Indigo now.
Has to be before time.
Yeah. Indigo is a tagline. Before time. Always before time. Afrin madam, can you share like how madam said the list of all the, or maybe the title of all the selected entries with ACG team?
Yes. Yes, ma'am. Yes, ma'am. I'll share it with them.
So beforehand, they just have a quick look at just the title it would be, right?
Ma'am, for oral solids, I've already shared with them. Do you want me to share for AI and other segments?
You can share. Yeah, Yeah.
Okay. Okay, ma'am. I'll share.
perfect. Yeah, all right. Just two more minutes and we go ahead. Yeah. Look, okay, sir, Afrin madam, let's start as 10:10.
Yes, ma'am.
Go ahead.
Guys, stop that. पहले पीपीटी करना है
Yeah, go ahead.
ओके क्या करना शुरू करें
हाँ, sir.
What do you do? Both of them ask when can we start? And then you start.
Yes, Lokesh sir, your voice we can hear. Yeah, start.
A very good morning to everyone present here today. I, Juveria Dosani,
And I, Avni Jain,
along with our entire SRC team, am delighted to welcome you all to the day 2 of the 6th Student Research Congress 2025, organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Autonomous, and co-hosted by the University of Mumbai, with the continued support of our esteemed industry partners, ACG Worldwide and SciTech Center.
yesterday we witnessed exceptional presentations, meaningful discussions, and a vibrant exchange of ideas that truly reflected the spirit and innovation and research and excellence. As we step into day 2, we carry forward the same momentum, enthusiasm, and curiosity.
Today promises to be an enriching and intellectually stimulating experience, and we are delightful to begin it with our welcome advocate, a visionary leader and an inspiring professional.
Yeah, continue. You're back.
It is our privilege to introduce Dr. Subhashi Chakrabarti, General Manager, Global Product Management at ACG Capsules. Dr. Chakrabarti holds a PhD in Pharmaceutical Sciences from IIT BHU and brings with him over 17 years of rich experience across global pharmaceutical MNCs, contributing to R&D, business development, and product innovation. Currently, he spearheads global product management initiatives at ACG Capsules. leading breakthrough capsule technologies from concept to commercialization.
Dr. Chakraborty has previously served at Eonic India Private Limited for 10 years in roles ranging from regional sales director to head of technical services for the Asia South region, supporting major pharma companies in developing cutting-edge injectables using biodegradable polymers. He also spent 3 years at Lupin Research Park, contributing significantly as a formulation development scientist.
His impressive body of work includes over 40 international scientific publications and 2 book chapters, and he has been honored with numerous awards, including the IIT BHU Alumni Association's Publication Scholarship Award and for 2 consecutive years, and the Best Alumni Award for his career achievements. Beyond his scientific expertise, Dr. Chakraborty is also an accomplished author of 3 self-help books, Happiness Decoded, Womb to Grave, and Touching the Core, all available globally. He is admired for his passion for mentoring young minds and contributing meaningfully to the academic and professional growth of students. We are deeply honored to have him with us today.
So I take this opportunity opportunity to welcome, Dr. Subhashish to our virtual platform of SVKM's BNCP. You have always been an inspiration whenever you have come to our institute and interacted with students. Students get motivated, inspired, not only for the pharma profession and the careers which you talk with them, but overall how to lead in their life. That's something which you always discuss, interact, and motivate students for. So we sincerely thank you for being with us today. And I'm sure your insights in healthcare and innovation is going to definitely make an impact on students' career and thought process. So over to you, Dr. Subhashish, please.
Thanks a lot, madam. And I think it's a pleasure. The pleasure is mine, and it's an honor to be a part of this. Not only this, but all the events that BNCP hosts. I think it's a wonderful institute going global and creating landmarks, day in, day out. Okay, so I think it's all it's an honor for me. So having said that, today is the day for all the students who are here. And first of all, I would like to congratulate each and every student here, because I don't know what's the response going to be, but you have already achieved something, a milestone already created by being able to be a part here. You have already started participating into a bigger forum, which itself is a milestone. So congratulations for that. And I think we're going to have a very healthy discussion today. Not only your presentation, but we'll talk, okay? Because it's all new for us, new for you, okay? So we'll evaluate many things, and probably one research will lead to the other research. Okay, so having said that, yesterday you got to hear a lot what's going on across the world, from the chief guest, Dr. Parizad, whom I closely work with and get inspired from the keynote speaker as well, Dr. Santosh, and they spoke a lot about what's going on across the world in the field of pharmaceutics and beyond that, okay? So that's an external world, okay? Now, what I'm going to talk about is something which is going on within me, okay? And believe me, this is nothing to do with what's going on across in the outside, but certain questions which has started coming up and, questioning me whether everything is fine, or is there something that we need to think of, okay? And and that's what I'm trying to share with you today, okay? So I hope my screen is visible.
Yes, visible.
Can I give a...
Yes, sir, you are visible.
Perfect. Okay, so let me first submit that when you talk about AI, and innovations, AI leads to innovation for sure. But a few years back, maybe two to 3 years back, I was in conversation with one of the professors. Okay, I don't want to take her name, but then Yeah, before I give or narrate the story, it's important to say that, and it's a confession that I didn't believe in AI. Okay, I felt like if I get adopted to AI, probably it's going to take my intelligence away. Whatever I have a capability of utilizing my brain. Probably that would stop. Okay, but when I started interacting with the professor, despite the fact that I shared my thoughts against AI. She was quite, humble to say that no, AI is actually helped her, and the way things have improved, the speed, the quality, efficiency has tremendously improved. Okay, and that is where when I came back home, it gave me a big kick. Okay, and the reason being that I felt like, am I missing something? Okay, the entire world has started adopting to it, and if I don't do, will I miss the bus? Will I regret in the future that I did not uh, match up to the technology which is available today? And I start questioning myself in the later years. Okay, and that is where I think a transformation happened, and that the first day where I downloaded the AI tools and started working on it. And to my surprise, it was like a heaven for me. I felt like now I can utilize. I mean, for sure there is a certain level of intelligence or the thoughts which is there within me, but that can go 10x by using AI. Okay, so having said that, I started utilizing the tools on a day-to-day basis, and then I could see the difference which started happening. It was a transformation for sure. I mean, nothing less than a transformation. And then but yes, there's always a scientific element which is always there, being a person from a scientific background, where that if I try to give a metaphor here, for example, a knife. Okay, it's good for multiple things. If you use it the proper way, it can make your life, but if you use it in the wrong way, it can take your life. Okay, and that is where I started thinking in different ways and try to balance my thoughts around it. Okay, so what I'm going to put before you today is, I think, the most simplistic presentation which I've created till date for the most complex, topic, which is artificial intelligence. And I thought if I can redefine it based on my thought processing. Okay, so let's go through it, and probably you would enjoy going through it. So the first question, and this is all what you want to see, only the questions, the answers either I would give, or you can think of yourself and you can take a call of what would be the right answer, as per you. The first question is, are we prepared to handle AI? Okay, now AI is something which dumps information like anything. The speed, the volume, of information that you get is tremendous. I think it's overwhelming to get information for any single topic that you talk about. Okay, but then are we prepared to handle? Okay, if we feel that that's intelligence, are we intelligent enough to handle that speed, to handle that quantity, and also the quality in terms of interpreting that data into something which is meaningful? Okay, and that is where I feel that if you are able to go deep. Okay, so I'll try to correlate two things. One is the width and the depth. Okay, so probably the AI is giving you the width of information, but it's you who has to find the depth. Okay, and that is where people get segregated between two forms. One is a specialist, where you go into the deep, whereas a person becomes generalist when you just jack of all trades but master of none kind of stuff, where you have ideas about multiple things but you are not very clear about what is the depth of something, where you can call yourself as an expert or a specialist. Okay, question to ponder upon: are you able to handle AI? Okay, so I think if you're able to understand this, probably you kick yourself to say that no, I have to also pull out myself as a human being to mature enough, in multiple ways to be able to handle the speed and the volume that AI is dumping on us. Okay. Let's go to the second question. Are we getting the right response, okay, from AI? Okay, so when you probably, you guys are presenting certain things and must have used AI for some reason or the other. Okay, and in that case, you are looking for certain responses, but where is that response coming from? The response is coming from the questions that you ask. Okay, which is a very scientific term used in AI, that's prompt engineering. Okay, now it all depends upon what quality of prompt engineering are you using. Okay, so when if I try to segregate that means that are you using the right questions. Okay, at the same time, the answers that you get, you also need to understand, is a reflection of your thoughts, because the questions are from you. It's not the AI which is asking the question, it is you who are asking the question. And if you ask the right question, okay, you get the right answer. And you can only ask the right questions when you are clear what you want. So basically, the response that you get is a reflection of you as a person, you as a personality, you as a caliber, you as an intelligent person or an expert. Okay, so think from that angle, that are you clear about what you want? Okay, now let's go to the third question. Okay, so I'm going to ask 5 questions. Okay, keep it short, otherwise it will be overwhelming because there are many questions. But I want to bring out the top 5 questions which I am concerned about. Okay, now the third question is: does the AI response needs improvement? So this question comes in when I see people around, just picking up the responses and throwing it off. Okay, without scrutinizing it. Now you need to understand that when you get a response and it's dumped as as an information to you, you need to segregate that. Okay, you need to refine it. Now, when I'm saying refining, it means you have to titrate it or customize it depending upon the circumstances, depending upon your audience. Okay, now if you are trying to put it across to an R&D audience versus a commercial audience, the answer has to be completely changed. Okay, it has to be refined, it has to be polished accordingly. Okay, sometimes the responses that you get is extremely complex because it's just a dump of multiple forms of information, and I've seen this happening in another event where a small problem, was presented in such a way that it became too complex even to understand the problem. The problem statement itself became so complex that it was difficult to analyze and come down to a conclusion. So decision making becomes extremely difficult if the response is too complex to handle. Okay, and that is where the human element comes in place to understand the answer which I'm trying to convey, the message which I'm going to convey to my audience. Okay, is simple enough to be understood and easy enough to take a decision. Okay, and I'm sure that today when you guys will be presenting your posters, your work, your research, I'm sure that you'll try to keep it simple. Okay, because it's you who is an expert in the area, but your judges, your audience, okay, may not be that expert in that particular area. So it's your, if you can succeed, or you would be the winner if you're able to convey the message in the most simplistic form, where probably an expertise is not required. Okay, so that is where improvements are extremely important. You cannot just pick up a response and put it across. Okay, there has to be a certain level of data refinement, data exclusion sometimes, or maybe addition. Okay, and that is where a meaningful data can be presented, and it could be worth presenting. Now let's go to the fourth question. Okay, and I think um, this is related to something which we see in and out today, that AI is overpowering us. Okay, but it's a question: is it overpowering us? That are we dependent on AI for each and everything? For the even the smallest thing, are we dependent on AI to support us? Okay, and I'm asking this question because I see many of my colleagues who probably are kind of they've given up thinking, okay, which is one of the most defining factor to create human beings. Okay, and if you're not able to think and you're completely dependent on AI to provide you any each and every information, I think there is a concern. Okay, we are losing the human element there. Okay, so are you addicted to AI? Are you overusing it? Okay, for maybe something that you could quickly come across, maybe take a decision. Okay, but you're still dependent on AI. So what could be the implication of such a situation where you are completely overpowered or addicted to AI? Is that your decision making power is going to go get compromised? Because when you want to take a decision, okay, for example, in a meeting or while discussing with your peers, colleagues, and stuff, in that case, you cannot just open a AI and say that, okay, let me check what the AI says, and then I will probably come to a conclusion. Okay, so there is no human element there. There is no prioritization, and there is no sentiments involved. It's only data which overpowers you. Okay, so think about it. Okay, and if this is what you're going through, I think it's good time that you start thinking and take a decision. And the final, I think there will be a lot of eyes which will go up that I'm asking this question: is AI reliable? Okay, I'm sure that we just blindly trust on the information that we receive. Okay, a part of this reliability also depends upon if you're asking the right questions. But yes, in some cases, I've seen that when I have asked for certain responses, I got certain things which were kind of confusing, which I reverted back saying that, ChatGPT, you are confusing me. ChatGPT said that, yes, sorry for that confusion and let me refine it. And then I started getting the right questions. Okay, so such kind of a situation I have encountered, and that gives me or raises the concern about the reliability of AI, which depends upon, again, the question you're asking. At the same time, it was quite a surprise for me while discussing with one of my colleagues in the office couple of days before, where there is a humorous campaign going on by Cadbury 5-star marketing campaign. Okay, and that is it's a very interesting information which I received, that that just for the sake of fun, they are trying to get into a zone of MAMA, which is make AI mediocre again. Okay, so if you want to understand that, you can Google it out and find out what's going on. But the intent here is, although it's a humorous intent, they want to, dump some nonsensible, information and reduce the reliability of AI. Okay, the accuracy level of AI. Okay, so this may be a very small, it could be a drop in the ocean as far as data is concerned across. But if such kind of this basically raises a question that, all the AI responses are dependent on the data which is available or which has been fed across, okay, across decades together. Okay, and today we are dependent on the data, quick access to those data, and therefore, we need to be careful about, and as we move ahead, we need to be careful about the reliability, recheck whether the information we are receiving is right, or there needs to be certain amount of titration which needs to be done to check or validate the data received from AI. Okay, so that's where I try to redefine AI, and for sure, it is called artificial intelligence, but I would say that it's an augmented intelligence, because it's not here to, to replace us, but to complement the human element of intelligence, and that is where I'm trying to bring in the element of redefined AI as augmented intelligence. Okay, so this is my final slide, and I'm again, putting you to work to think that AI is not here to replace your intelligence; it is here to amplify it. The true power of AI lies not in the answer it generates, but the question you dare to ask. And if AI can give you the speed, it's only you who can give it a purpose. Okay. And finally, the data will guide you, but it's only you who can turn it into a decision. So, guys, it's time to put your brain to work, not completely depend on artificial intelligence. Okay. Ensure that you as a person is present and you make it happen. And I'm expecting the same thing in today's in the event today in your presentations, where I would depend on the data from AI, but I would look towards you and your decision making, your thought processes, and try to find you in this entire conversation. Okay, so that's all that I had to share today. I hope you enjoyed it, and I look forward for a meaningful discussion across the day. Thank you.
Thank you very much, sir, for your insightful and inspiring welcome address. Your words have truly set the tone for day 2 of the 6th Student Research Congress 2025. We deeply appreciate the thought and vision you have shared with us this morning.
We request everyone to kindly settle in, ensure your microphones remain muted, and prepare for yet another day filled with learning, collaboration, and discovery.
Once again, a warm welcome to all. Let us officially commence day 2 of SRC 2025. We will now continue with the PhD category oral presentations, which will proceed in the room 3.
All presenters for the Hackathon Oral Solids category are requested to kindly join room 4.
Yeah, just a second. So before we officially start with the AI and PhD program hackathon, let me officially thank Dr. Subhashish for setting the tone for today's both the session, where he will also be one of the judge. So thank you once again, Dr. Subhashish, on behalf of BNCP. Over to you, Team BNCP.
All presenters for the Hackathon Oral Solids category are requested to kindly join room 4. Similarly, presenters for the Hackathon Artificial Intelligence category are requested to remain in the same room.
We also extend a warm invitation to all viewers and attendees to join the respective rooms and witness the innovative ideas being presented.
For your convenience, all videos from today's and yesterday's session will be available on our official YouTube channel, SVKM BNCP, and on the SRC website, bncp.com, after the event.
We also look forward to your presence at the valedictory session scheduled after 2:30 p.m. today.
The meeting links for each room have been posted in the chat box. Kindly refer to them and join your allotted sessions.
Thank you.
Thank you, and let's meet into the respective area. We can, the respective rooms, we can go. In the chat box, there is a link. So let's start.
Before we start, we take a 5 minutes quick break and we start with that session.
Good morning, everyone.
I, Mahi Rizvi,
And I, Ikra Khan,
are delighted to welcome you all to a very special and newly introduced segment of the 6th Student Research Congress 2025, the Hackathon Artificial Intelligence. This year marks a significant milestone as we expand our scientific horizon with this dynamic, innovation-driven hackathon. It is an initiative proudly curated by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Autonomous, co-hosted by University of Mumbai and supported by our valued industry collaborators, ACG Worldwide and SciTech Center.
the hackathon is not merely a competition. It is a dynamic arena where intelligence meets innovation, data meets precision, and technology transform idea into impactful solutions. To set the tone for today's session, let me share a thought that perfectly captures the essence of today's challenge. Artificial intelligence is not about replacing human thinking, but about amplifying it. This powerful idea reflects the true spirit of AI hackathon: analyze, innovate, and transform. In the Artificial Intelligence Hackathon, participants move beyond conventional learning. They engage in real-time problem solving, data-driven reasoning, algorithm thinking, and practical applications of AI to address pressing challenges in healthcare and pharmaceutical science. This platform encourages you to blend scientific insight with computational intelligence to design solutions that are smart, scalable, and meaningful.
To all the teams participating today, your presence here reflects your curiosity to explore emerging technologies, your courage to experiment with data and models, and your commitment to shaping the future of healthcare through intelligent innovation. May this hackathon spark bold ideas, sharpen analytical thinking, and inspire solutions that redefine how technology can enhance patient care and pharmaceutical research. We wish all the participants our very best. Let intelligence guide innovation,
And may the most impactful idea emerge victorious. We are honored to be joined by such distinguished experts. Their guidance and experience greatly enrich today's proceedings. Dr. Abhijit Pandey, Dr. Anup Kishore, and Dr. Atul Sethi. Dr. Abhijit Pandey is a seasoned pharmaceutical formulation scientist with over 10 years of product development experience, specializing in complex and long-acting injectable drug products. He currently serves as Manager, R&D Competitive Intelligence at Novartis Healthcare. His expertise focuses on designing innovation strategies and control technologies for complex injectable, including new chemical entities, NCEs, new biological entities, NBEs, and oligonucleotide. He has also led strategic competitive intelligence initiative, supporting R&D teams through scientific due diligence and technological landscape assessments. Dr. Pandey also holds doctorate in pharmacy from Maharaja Sayajirao University of Baroda, conducted in collaboration with Baba Atomic Research Center, BARC, Mumbai. With a focus on radioligand therapy, he completed postdoctoral research at Manipal College of Pharmaceutical Science, MCOPS, in the Department of Pharmaceutics and was awarded the prestigious EMBO, European Molecular Biological Organization Fellowship, to pursue further postdoctoral work in biophysics and material science at the Medical University of Greece. He has been organized among the top 2% scientists globally for 5 consecutive years. Including 2025, Dr. Pandey has received several esteemed fellowships, including those from AICTE, UGC BSR, Commonwealth, ICMR, BRNS, DST, and EMBO. His research portfolio includes over 100 publications in high-impact international journals such as RSC, ACS, and Nature, accumulating a cumulative impact factor exceeding 500. He holds more than 5 patents. Beyond his scientific contribution, Dr. Pandey is actively engaged in sports, cultural pursuits, and animal welfare. We are truly honored to have such an accomplished professional with us.
Thank you, Dr. Abhijit, for being here. we sincerely appreciate that. I know in spite of lot of busy schedule, you accepted our invitation. So, and it's been a pleasure seeing you once again over here. It's been almost a year, I think. We met last year, and thank you very much for being here with us on our virtual platform. And look forward to a lot of insightful discussion from you. Thank you very much.
Thank you, ma'am.
Thank you.
Today, we also have with us Dr. Anup Kishore. Dr. Anup Kishore is an accomplished academic and researcher, currently serving as an associate professor in the Department of Pharmacology at Manipal College of Pharmaceutical Sciences, Manipal Academy of Higher Education, MEHE, Karnataka. He holds the important role of coordinator, Center for Digital Learning, MAHE, contributing significantly to innovation in teaching learning practices. Dr. Kishore completed his PhD in pharmaceutical sciences from Manipal University. With over 15 years of teaching and research experience, he has been actively involved in undergraduate, postgraduate, and doctoral training in pharmacology. His research span neuroscience, diabetes, metabolic syndrome, inflammation, and immunity, cancer, and cognition. Dr. Kishore has successfully secured competitive research funding from prestigious agencies including DST, Fast Track Scheme for Young Scientists, VGST, CISEE, Establishment of an E-Learning Center, ICMR, Drug Therapy for Glioblastoma, along with multiple industry-sponsored projects. He has guided 20 postgraduate students and is currently supervising 6 PhD scholars. Dr. Kishore has authored around 50 research publications and has presented his work at national and international conferences, reflecting his strong research footprint. With his blend of pharmacological expertise, digital learning leadership, and research excellence, Dr. Anup Kishore continues to make meaningful contribution to pharmaceutical education and translational research. We are truly privileged to have him with us today. His experience, insight, and guidance add immense value to our proceedings.
Thank you, Dr. Anup Kishore, for being with us here on our virtual platform. I'm sure your insightful discussion would definitely help students today. Yeah, over to you, students.
We are also pleased to introduce Dr. Atul P. Shetty, Head of Department of Quality Assurance at SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai, with 19 years of teaching and research experience. He holds a PhD in pharmaceutical sciences, NMIMS, and specializes in analytical method development and validation, nanocarrier-based drug delivery system, and solubility enhancement of poorly soluble drugs with strong expertise in advanced analytical instrumentation. Dr. Shilje has been featured among the top 2% scientists in the world, Stanford Elsevier 2023, and has received multiple recognition, including the BNCP Shining Star Award 2023 and allocates for mentoring award-winning student research at Avishkar. He has guided 25 plus M-Farm projects, completed industry consultancy projects, and has a strong publication record with H-Index 20, I-10 Index 30, and 1,360 plus citation. We are honored to have him with us today and look forward to his valuable insights. Thank you for your gracious presence and for dedicating your time to this event.
Thank you, Dr. Atul. I'm sure 3 of you definitely make a lot of impactful discussion and interaction with students. Thank you, everyone.
My pleasure. Thank you.
I request all the participants to carefully go through the instruction displayed on the slide. With the permission of our esteemed judges, we are ready to begin with oral presentation competition for Hackathon Artificial Intelligence. Our participants and audience are eager to experience the innovative and impactful idea presented today. Also, I kindly request the judges to confirm that they have received the scorecards.
Yes, we are received.
Yes, sir.
Yes, I have received.
I would now call upon code 002 to switch on your cameras. We will be presenting your video now.
Yeah.
Code 002, are you there in the meeting? Code 002, please confirm that you are in the meeting. Please unmute AI 002. Please unmute yourself.
A very good day to everyone. My ID is AI002 and today I'm going to be presenting DeepCure. This is a tool that could be used for AI-driven antibody drug conjugate ADC designed for cancer therapy. Now the actual problem statement that I'm addressing here is the fact that these traditional anti-cancer drugs are associated with toxicity. They don't have a really targeted action against these cancer cells. ADCs is a type of therapy where you have an antigen which is overexpressed on your cancer cell. You're going to be deciding on an antibody that is particularly targeting that antigen, and this antibody will act as a guide for your payload, for your actual anti-cancer drug. And developing these ADCs is a costly and time-consuming process. There's a whole lot of trial and error involved. You have to choose the right linker, the right payload, and all of this is really hectic. This is what my tool is able to do effectively. Why can't we just use the technology that's already available? The existing solutions are not up to the mark because traditional molecular docking, simple machine learning models, and component-specific analyses, all of these methods, neither of them provides a holistic view which is necessary when you're developing these ADCs, and something that my tool does have, as we'll see. The proposed solution over here is divided into three major steps. The first one being protein representation learning. We'll be looking at the antigen and the antibody and studying them based upon tools like Prot5XL, which is a protein language model. We'll look at the sequence of the proteins in the antibody and the antigen and determine what sort of function it is probably going to be having. Tools like AlphaFold 3 give us a 3D view of this entire structure and give us an idea of how the proteins are going to be folding, which helps us figure out what sort of structure and function they're actually going to be shown. The second step would be creating a multi-dimensional graphical representation of the linkers and the payloads that are present over here. So now these are extremely important because their vicinity is going to be telling us how similar particular linkers and payloads are to each other. And the third and the final part of my tool is going to be a unified multi-input AI model. And the entire purpose of this model would be to provide a sort of fusion network where it will take in all of this different type of data, develop patterns, and use this to create hopefully accurate prediction. The major aspect of novelty in this project is the fact that I'm using a holistic view in making predictions of these ADCs. I'm not just looking at antigen and antibody interactions. I'm also seeing whether or not the entire structure is actually integrating into the cell and then destroying it. So all of these small features are extremely important, and that is something that my tool does include. There are a bunch of different molecular fingerprints that are used to represent chemicals, and each of them have their advantages and disadvantages, and my idea was to make use of each of them to get a better idea. And this image just shows the integration that I was talking about. Here's a quick visual representation of how this project would work, collecting all of this ADC data, processing it through all of these tools, and training a multi-layer perceptron on all of the data that I've used, and then filtering out to give prediction. This is what the data set, the initial data set would look like, and this is the workflow that I'll be following using this data set to train the neural networks, and then providing predictions essentially. I see this tool being used by physicians to provide personalized treatments to their patients. For instance, they'll be looking at the immunohistochemistry report of a patient and finding out that the HER2 positive cells are overexpressed in the breast cancers of a particular patient, and use this information to provide the right ADC to them. It can be used by the pharmaceutical industries R&D wings, because they have like a list of different different ADCs that they want to test, and they can use this tool to narrow it down to 10 or 15 more prioritized ADCs which are expected to be more effective. Because we're going to be creating a graphical representation of these different ADCs, we can use it to determine particular cluster. If as a pharmaceutical company, I know that there are a few of my drugs that are effective in a particular cluster, I can be confident enough that the other drugs in that cluster are also going to be effective. These are a list of different ADCs that are already being used, and these are some of the ADCs that are being used in different types of breast cancer. There is definitely a market out here. From personal experience, working in an pharma consultancy company in Pune as an intern, I was able to find out that while initially they had demand from these large pharmaceutical companies for an ADC activity prediction tool, the small or medium companies used to never really ask for such a tool. But in recent times, even those companies have started asking for this, and the highly expected reason for this is that 15 to 20 years back, so many of these companies had put patents on their ADC designs, and now these are going to be coming out into the open market, and it is going to be a gold rush. There are a few ideas that I have on how this project could be scaled in the future, but one of the most exciting ones that I'd like to discuss today is that right now the tool essentially takes information about an ADC and gives me a prediction about its efficacy, but I'd like to flip the arrow wherein I provide the tool an antigen and it gives me a particular effective ADC that could treat a cancer cell with overexpression of that antigen. Essentially a sort of generative AI tool of that. The proof of work is over here. This is how the user interface would look for this tool, and this is how the sort of the prediction that it would. And another thing that I'd like to discuss is that of validity, wherein these tools can be validated using data that has not been taught to the model already, and based on their predictions we compare and find out how effectively it is working, and my tool is able to get an AUROC score of in the range of 91 to 90. While this score is not enough in terms of industry standards wherein you need 9%, this is something that can be worked on if the data set is enlarged. And the plan that I have for enlarging this data set is to provide license to this tool to different companies, and if the companies, which most likely they won't be willing to provide their data, they can use their data in their site premises to train the model, and this will allow them to get a more personalized prediction and allow me to get a better and stronger. And in this entire scenario, the data never leaves the premises of the company. And that was all from my side about DeepCure. These are the references that I've used in this project, and thank you.
Thank you, Code 002. I now invite the judges to ask their question to the respective presenters. So
Am I audible now?
Mute.
Yeah. Am I audible now? Am I audible now?
Yes, sir.
Yeah. Is this just a concept or have you actually worked on this?
So I've worked on a prototype of it. The way that the entire process works is that I have an initial data set, but that just has 2000 ADCs in it. And
Okay.
I've trained the
Yes,
model
yes.
on it as well.
Yeah, sorry to interrupt because there are other judges as well to ask this question. So Second thing is, which part of the pipeline are you targeting? Is it the target, the linker, or the payload? What is it that you're targeting?
It's the initial phase, I believe, before preclinical trials, wherein a company which has a bunch of different, like if they have 1,000 different ADC molecules that they are not sure if they should go ahead with testing it, they can come to my company and then they can use my tool to list out only 15 or 10 of them, which are
No,
more
no.
likely to be
Are you
effective.
looking at the linker, or are you looking at the payload? Are you looking at the target? What are you looking at?
We are
That is the
majorly
question.
looking at the payload, but why I can't answer it exactly clearly is because I'm also looking at the antigen, which in this case is the target as well. But my payload is the main thing that I'm studying over here.
Okay. 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 Thank you.
Okay, I've got one question that when you are training your data set and you say that you're working on payload,
Yes.
how will you train your data
Yes.
set with a new payload which you have no information about? Because all the DCs are new and the antigen will be new,
Okay.
keeping in mind the latest development. So how are you going to train your machine?
So the data set which I'm training my machine on has, you can imagine it like there is a list of different ADCs, and there is another list that the labels which are telling me whether they're active or inactive. And how have I decided whether they're active is if they are in the market or if they're in phase 3 and 4. If they're in phase 1, 2, or even in phase 1, 2, 3, 4, then they are considered active. And the ones that are in the preclinical stages or they're still not in the clinical trial phase, they are considered inactive. So I'm
Okay,
training my model looking at these labels and trying to use all of the different features that I mentioned, Proti, XL, ESM5. These are giving me the features, and my labels are whether it is active or inactive.
so it won't be hold true for new NB. Like if I want to have design a new drug and new antibody, which has got no data set, so you won't be able
Yes,
to help that with drug screening. You can only help with
I
the
will be able to help
phase
that.
one. Okay,
No,
great.
sir, I
Thank
will
you.
be able to help that. What I'm saying is that my data set would have the labels, and the consumer, the person who's using the tool, can bring in a new ADC. And my tool has learned the patterns from the data set that I've provided. That these patterns are seen in ADCs which are normally active, and these patterns are seen in those that are considered inactive. So in the case of your new ADC compound, it would try to figure out what these patterns are for your particular ADC compound, and then give a prediction.
Okay, great. Thank you.
Okay, you have mentioned that you have carried out validation of the study, right? Am I right?
Yes, sir. Yes.
Validation, but you also mentioned that the validation scores are not satisfactory. So, are you going to
Correct.
validate, or how are you going to go ahead with that? Because
So the
whatever,
process of
yeah.
validation and this. Sorry, sir. If you
Hmm.
want to continue your question,
No, please
please
go
go ahead.
ahead. Yeah.
Yes. So the process for validation and making any sort of machine learning model is the process that I followed over here. And in previous tools that I've studied that deal with drug activity prediction, there are many other ways that we can use. There is precision recall, and I have just stated AUROC over here, which is under the expectations of what would be required in the industry. Now, the way to deal with this issue would be to create a better data set. Uh, Considering that I have only used 2,000 ADCs in my data set, that score is satisfactory. But an industry, a company would not be happy with it. For me to increase that, I have a plan wherein I would be
Hmm.
providing the model at the current stage, for example, to a company, and this company could train it on their own data. If they're not willing to provide me the data, they can do it on their site premises itself. This will help them because
Hmm.
it gives them personalized it gives them better predictions for their future ADC compounds. And for me, it's better because I'm able to train. And this is a system called federated learning that is even used by many AI companies today as well.
Okay, all right. Thank
Yes, so
you.
that would help me.
Yeah.
That would help me get a better model. Yes, thank you.
Thank you, judges, for your valuable question. I now call upon code 004 to switch on your camera. We will be presenting your video now. Code 004, can you confirm that you are in the meeting?
Hello everyone, greetings of the day. Today we are here to present Nomosi, an instant relief anti-nausea inhaler. Our presentation code is AI002. We belong to the category of Hackathon in theme Innovations and AI in Healthcare. We are presenting a traditional herbal inhalational therapy rationally optimized using AI for formulation precision, safety, and rapid onset. Now, talking about background, the problem. So before we begin, let me just ask you something. Have you ever felt like nauseous during traveling, maybe a bus ride or on a curvy road or in a friend's car with too much perfume in it? Exactly. Nausea doesn't wait for permission. It just hits at that moment. As you can see, prevalence of nausea is very high. 35% in gastrointestinal disorders, 25% in CNS causes, 15% in drugs, toxins, and chemotherapy related nausea, and likewise. So the numbers are definitely not negligible. We need to work on that. We need a solution which is instant acting, which is drug free, which is safe to use. There are existing traditional therapies like tablets and syrups. They work, but they act slowly. They have side effects. Tablets have side effects like they cause drowsiness, and they are not practical in sudden attacks. So there is a clear need of a solution which is instant acting, which has minimal side effects. We have heard from for centuries that volatile oils like ginger or lemon is used for treatment of nausea. So on clinical research, we found that volatile oils like ginger oil, lemon oil, peppermint oil, and so on are used as anti-nauseatic, are used for its anti-nauseatic effect, and this is clinically proven. So we have formulated a inhaler named Nomosic, which is instant acting. It has no almost minimal side effects. It is highly portable and user friendly. It is safe to use. It is drug free and it is very affordable.
Now coming to the rationale, some questions may arise in your brain. Why an inhaler? Why we choose an inhaler as a formulation? When you inhale something, it reaches to your brain within 0.2 seconds, which is very fast. So our inhaler provide rapid nasal absorption. It is non-invasive therapy and it is very portable and patient-friendly. The second question may arise, why herbal volatile oils? As they act via olfactory limbic pathway, that is, they produce very rapid effect. Second, minimal systemic exposure, so no more deal with the ADRs and the toxicity. The third thing is they have well documented safety. Volatile oils are used traditionally and their safety is well documented. Then the third question may arise, why AI? Here, herbal formulation traditionally depend on trial and error basis, but with AI, it comes with the precision. Traditionally, formulating the herbal formulation is like cooking without a recipe, little of this and little of that. But think of AI like a chef who know every spice of the world. With the help of AI, we are able to decide the right oil, the right combination, the right concentration, and we also come to know which things we need to add, which things we should avoid to add, and in what combination we should add, how to preserve that oil. The next part is innovative role of AI in this formulation. So we have used AI at different level of development of this formulation. The first thing was after identifying problem statement, we need a solution. To find a solution, we need to do literature mining. For literature mining, we have used tools like PubBird, SciBird, Google Scholar Lab, which help us to go through thousands of research papers and to find best thing which we need for our formulation. That is, oil, the combination, the concentration, everything. Then second is the predictive modeling. We have used the tools like random forest and gradient boosting, which help us to find the right combination of oil we should use in our formulation. And the third thing is the optimization. We have used Bayesian optimization and sometimes genetic algorithm, which help us to identify the ratio in which we should use our oil to produce maximum effect and minimal irritation of the olfactory. The second most part is the methodology, how we formulated this formulation. The first thing was the problem identification. This was a real life problem identified by her, identified by me during our traveling to our hometown in vacations. We see everywhere around us, and therefore we find our solution by doing literature reviews and whatever literature mining. Then we form a formulation strategy, then prototype development. As you can see in this image, this was the first prototype, and this is in my hand. It is the second prototype, which is at the TRL level of 3 to 4. That is, prototype is well developed. Now it is under lab level trial, and we will proceed with next TRL level, and so on. We are planning for the clinical trials. This diagram explains how it works on your brain. You just have to open this, inhale it like a normal inhaler. It act on olfactory lobes. From this, it act on a chemoreceptor trigger zone and suppresses the signal of nausea and gives the relief. In the picture given at the bottom, here we have explained how we formulated this formulation in our lab. The next part is the result. As you can see in the green graph, it shows the percent nausea reduction by the different oil oil. As you can see, peppermint oil reduced 44% nausea, ginger oil reduced nausea, but when they are used in a combination, peppermint plus ginger, they produce is more beneficial. For business expansion, we have B2B, B2C, and B2G model, because it is a product which has wide scope. So we can cater traveling agency, patient, pregnant woman, hospital, healthcare services, and many more. So there is no doubt for its scope. The conclusion. So concluding,
Behavioral. What if it is physiological, like due to...
And then you're supposed to click on allow.
Do you think you will make a profit with this? Will it be sustained?
AI-driven and cost-effective. In short, about the diabetes, that as everyone knows, that
Dear judges, can you give us a minute, please? I think there's a little network glitch here. We're just trying to fix it. Just give us a couple of minutes, please. Thank you.
diabetes is the major issue
Hello everyone, I will be speaking about my proposed idea, and it is regarding AI-driven, long-lasting subcutaneous glucose biosensor for continuous diabetes management. And key features about the product would be long-lasting, AI-driven, and cost-effective. So initially, I will be briefing in short about the diabetes. As everyone knows, the diabetes is the major issue, and the diabetes cases are increasing daily. And in future also the trend would be kind of the same manner, similar manner as provided in the type 2 diabetes global market report 2025. The expected growth rate to 2029 is around 8.50%. Moving on to the background about it. Diabetes mellitus is a chronic metabolic disease. There is a elevation of blood glucose level has been observed due to the impaired insulin secretion action or both. So the aim is to monitor the glucose levels to prevent any further complications such as neuropathy, retinopathy. Despite major advancements and being innovative, the current monitoring devices face challenges. And first challenge is the enzyme instability due to the enzyme denaturation or degradation with time. Short functionality lifetime, typically around 7 to 14 days. Biofueling means like due to the microorganisms that are in rapid occurs or arising from protein adsorption. Another is the high device consumable cost due to and which is a kind of discomforting as there's a repetitive or long-term insertion which can limit the patient acceptance. And the rationale. So address the limitation, the project proposed for the development of next generation subcutaneous patch being minimal invasive AI-enabled glucose sensor, which aim is to for the extended operational longevity of around 1 to 2 months, so that it can and it could be better in terms of performance over the current short-term sensors. So the design features in the three part like the subcutaneous filament, variable housing, and AI mode. Subcutaneous filament will be inside the skin around 4 to 5 millimeter, and the proposed design emphasizes on the covalent immobilization of glucose oxidase on nanostructure carbon electrode, which ensuring strong enzyme anchoring. And then there's a multiple protective and functional layers, including conductive polymers and selective membranes of DG. All this will be helpful in minimizing the interference and biofueling in physiological conditions. And whereas the AI-driven closed loop insulin delivery mode capable of interpreting the glucose data, like the spike or drops in blood glucose level, and to adjust the insulin delivery to prevent episodes of hyperglycemia. The wireless connectivity allows the data transmission and the display on smartphone application. The aim was to establish a stable and cost-effective biosensing platform, integrating nanotechnology, artificial intelligence, and incorporating model and reusable electronic components in the biosensor design that can significantly lower the cost. Moving on to the method. First is the electrode preparation, where the nanostructure can power with a carbon electrode. It could be carbon nanotubes. So basically, clean and activate electrodes by soluble wash and electrochemical treatment, electron enzyme immunization by covalently immobilizing the glucose oxidase on electrode, and on the protective coatings like applying the multi-layer coatings like the anti-fouling surface treatments. Another is the sensor operation. The sensor operates algorithmically by reading the electrochemical signals which are generated from the glucose oxidation, and the current and as the based on the glucose levels, the current will also increase. So basic optimization occurs over here to maximize sensitivity and selectivity. So another is wireless data transmission. This is integrating low power microcontrol with the Bluetooth and AI-driven insulin control. So and then the last performance testing. It includes characterized sensitivity, linear range, response time, to determine stability about up to 60 days, and to conduct an evaluation. So the results include sensor performance and stability. So under the E matrices, sensitivity should be around 40 to 100 microampere, and response time less than 50 seconds. Linear detection range should be around 1 to 30 mm, and R square greater than 0.98. So these are some of the features which should be taken into consideration and should be fallen into this category, kind of in the range. Selectivity greater than 90% signal retention against interference. So there's a graph which are plotted side by side, like left calibration curve determining linearity, whereas the right is stability curve, which shows activity retention. So here, major things which should be focused over here, like as it is AI driven. So under that, 76 to 80% timing target glucose range, prediction accuracy, RMSE is the root mean square error, which is kind of very much important. Should be around less than 30 mg per deciliter. Around 40% reduction in hypoglycemic episode, 35% reduction in hypoglycemic episode, and it should perform the other CGM systems. So whereas in the data analysis, first is the sensor performance analysis, and first is the sensitivity and linearity. There's here the plotting sensor current versus glucose concentration, and selectivity is comparing the response with or without interference. Whereas the stability monitor the sensor response over the 60 days and taking the actions, and there's the biocompatibility, which analyzes histological images and inflammatory markers from animal studies. Whereas in the AI-driven insulin delivery analysis, it basically looks about the glucose prediction accuracy, which can compare the predicted versus the actual glucose values. So calculating RMSE and mean absolute errors. So depending any errors, so it can be solved. So errors can be worked. So it gives a better idea. And visuals are also provided for each. And insulin delivery efficacy, it calculates the percent time in target glucose range. Most of the time it should be in the target glucose range, which is around 70 to 80 mg per deciliter. Also the pie chart for time and range and bar chart for episode reduction. So with this, I would like to conclude saying that there is a successful integration of advanced nanomaterial engineering by compatible polygons and artificial intelligence. Main thing should be over here focusing on the code and immobilization, predictive algorithms, being proactive rather than reactive, focusing on forecasting glucose fluctuations, and adjusting the insulin delivery accordingly to the therapeutic response, and being patient compliant and being lower in terms of long-term health costs. And modular architecture ensures scalability and adaptability. So these are the references. Thank you.
Thank you, Code 005. I now invite the judges to ask their question to the respective presenter.
Am I audible?
Yes, sir.
Yeah, sir, you're audible.
Yeah. So, this project that you have told, there are so many components to it. There is electrode preparation, there is enzyme immobilization, there is sensor, there is a wireless connectivity, there is AI, then there is AI driven. So AI is only a very small part of it. That is to capture the glucose concentration and preventing the signal drift. What I feel is each of the components that you mentioned, at least 7 to 8 components each of them are separate science by themselves. Do you think you will be able to do all these things? And it's like equal to some 7 PhDs. Okay, if you're going into that. And the AI is for a very small part, that is the drift. You should have concentrated on the drift, no, rather than making the entire device.
Sir, am I audible?
Yeah, you're audible.
Yes, sir. So basically, as you said, like there are multiple components. So yeah, it's a part of the BIOS website, but my main focus is over here is like to give more on AI-driven insulin delivery. So like based on the fluctuation which will happen in the glucose levels, it will detect that, and through the insulin pump, it will mainly target that and to maintain and to keep it in a normal range. yeah, I can understand. Like you just told key, like you should give more focus on the AI part, but it's like it's based on this like study model. I would later on focus on like giving more on AI, but it's kind of been AI, because it's like as I told you, it's kind of been a Bluetooth, low power microcontroller with Bluetooth, so it's been a little
Okay.
bit more accurate as compared to this.
Okay.
I've got one question. For how long do you think the device can work at once? Like for how many days?
So our target is accordingly for around for 60 days.
And how do you compete with the company like Abbott and Novo Nordisk, which already have body patch glucose monitoring system for almost like they recently launched few of the AI enabled where you don't even need to refill it. They have the sensor. You can select your iPhone or Apple Watch, click the timing when your drug leaves. It's done. So how do you differ from that part?
So basically, sir, as you said, like, yeah, they might be having the system, but what are the current, which are the sensors? The main problem is of regarding enzyme degradation, because of which it is not long lasting. It would somehow get degraded in 1 or 2 weeks, but
So 30
eventually,
days
mine
by
target
Novo
initially,
Nordisk.
yeah, but mine is around 1 to 2 months. I'm just thinking beyond like around going initially 1 to 2 months, but it's still like, which I would like increase. But the main focus I would be giving is like the enzyme degradation shouldn't happen, and that the problem of biofouling, which is being taken into consideration, should also be prevented with the multiple coatings. And yeah, as I also taken into consider that refilling what issue that there's no like, kind of thing. But in my case, I also been giving more focus that the refilling of insulin is being avoided at max to max, and how efficient and low cost I can make it.
Okay. You're working more precisely on the cost part.
Yeah.
Okay, great. Thank you.
Okay, I just want to get a clarity on your project. So what I understand is you have developed some device, right? For continuous monitoring of glucose.
Kind of.
Yeah, wireless device. Yeah. So have you developed any prototype of that, or it is just a predictive study? I just want to get a clarity on it.
So sir, it's a predictive study. I have not yet started with any prototype regarding this.
Okay. So use of AI is just like for it can be prepare for optimization, getting composition and evaluation parameters basically.
Yes, sir.
Okay. All right.
Thank you, judges, for valuable questions. I now invite code number 009 to switch on your camera. We will be presenting your video now. Guys.
My identifier number is AI009. In many parts of the world, access to fast, accurate, and affordable diagnostic tools remains a challenge. Disease linked to liver and bone disorders often go undetected due to the lack of early diagnosis. What if a simple piece of paper could change that? Hence, we came up with a prototype titled Affordable Paper-Based Fluorescence Sensor for Alkaline Phosphatase Detection, a game changer in point-of-care diagnostics. Current diagnostics method for alkaline phosphatase relies on expensive lab-bound systems that require skilled personnel. They are time-consuming, costly, and unsuitable for field or rural use. We present a low-cost, paper-based ratiometric fluorescence strip that detect alkaline phosphatase with high accuracy. Entirely built using commercially available materials and chemicals. These innovations cost less than 10 rupees per test and require no instrument or synthesis. The sensor works on principle aggregation induced emission. It means TP4C dye alone shows negligible fluorescence in solution because of the non-radiative process, whereas shows strong fluorescence especially upon aggregation due to the block torsional relaxation. High sensitivity, large stroke shift, and excellent photostability are the advantages of AI dyes over conventional fluorescence probe. We are using tetracatanic dye TP4C, catanic polymer PAA, and ATP, which is tri-negatively charged. And another role is ATP is a substrate of an alkaline phosphatase. Hence, ATP is playing dual role. A tetracatanic dye TP4C alone shows negligible fluorescence but forms strong fluorescence aggregate with a catanic polymer. A trianionic ATP breaks the aggregate formed due to the dye and polymer interaction, electrostatic interaction. ATP is, as I said, ATP is a substrate of the alkaline phosphatase. In the presence of alkaline phosphatase, ATP is broken down, restoring the aggregation and strong fluorescent signal. This reversible fluorescence turn off and turn on process form the basis of ratiometric sensing, where fluorescent signal turn gives reliable and self-calibrated response. The mechanism of paper-based assay relies on simple yet powerful drop casting method. We have used reagent sequentially, dye, polymer, ATP, and different concentration of alkaline phosphatase from lower concentration to higher concentration. Dye alone is negligible fluorescent, whereas dye and polymer gives highest fluorescence. Upon addition of ATP on dye-polymer complex, fluorescent signal quench or turn off. This quench signal will be our reference point for alkaline phosphatase detection. In the presence of alkaline phosphatase, ATP is broken down, reforming the aggregate and causing a strong fluorescent signal. We optimized and validated this system in buffer as well as 5% human serum. Then we translate entire mechanism into portable lateral flow assay kit, where one spot acts as a control without LP and the other spot which is acting as a sample for detecting unknown alkaline phosphatase present in the serum. The fluorescent color shift is visible and quantifiable. Hence, we can take photo, and this photo we can read through digital color analyzer software which is inbuilt in smartphone. Using calibration plot, unknown alkaline phosphatase level in the serum can be estimated accurately. This ratiometric change allows real-time and highly sensitive detections. Key benefits and features are all materials and chemicals are commercially available. So no requirement of synthesis. This kit is simple, equipment-free fabrication. Because we are using only paper as a platform for detecting alkaline phosphatase. It is ratiometric and self-calibrated because of the turn on as well as turn off signal. For fabrication, no ink or printing or paper modification is required. Other than that, we are eliminating lab-based alkaline phosphatase to a portable paper-based alkaline phosphatase detection. Under UV luminance, we can observe the spot develop from alkaline phosphatase, which is present from unknown concentration in human serum. Then with this spot, we can check this in digital color image analyzer, which is in inbuilt software which in the smartphone. This software read through RGB color code, and with this unknown concentration of alkaline phosphates, we can check and compare with calibration curve. Now, scope for the commercialization. Filter paper-based platform can be used in environmental, food, and disease diagnostics by adapting to different enzyme targets. Integration with the smartphone compatible module enabling real-time and user-friendly diagnostics. With the standardized quality control procedures, the system can be reliably scaled for bulk manufacturing. The clinical validation through extensive trials with the diverse patient sample will help establish its accuracy and reliability. The regulatory approval for securing essential certifications, including ISO and FDA clearance to ensure safety, performance, and readiness for market deployment. Certifications and award. This work is appreciated in Gandhian Young Technology Innovation Award in 2024. In the summary, we are developing portable filter paper-based alkaline phosphates kit which is ready to use. Thank you so much.
Thank you, Code 009. I now invite the judges to ask their question to the respective presenter.
It was a good presentation. I have a few questions. Till what extent are you using AI? The first question. Second question is like, what is the timeline for detection, the overall time period, and why it is different to already available paper devices which is in market?
So for first question, we are using RGB color. We are going to take photo and with the smartphone camera as a detector we are using. Then we are using, there are different softwares which are already in the markets, like image or RGB detector like that we can use. And another option is we can directly upload this strip with the AI with specific prompt, and with that AI model, the trained AI model can directly quantify this color develop on the strip. Second question is what are the sorry sir, can you please repeat?
Available devices which is in market for paper-based test of the same thing.
So currently, okay sir. So currently in the market for alkaline phosphates detection, there are colorimetric methods, fluorescence methods, and some high costly instruments like surface enhanced Raman spectroscopies are there. They are used. Although they are sensitive, but they are costly and they require trained laborers. And in this filter paper based platform, there is no requirement of trained labor, and the color develop, which is within milliseconds or microseconds, maybe we can say that, means after dropcasting this sample on the paper, with the help of dye and polymer, the color is developed instantly. So this is a benefit. And there are other kit which is in the market. Although they are sensitive, but they are time consuming and not able to because of the what we can say.
Got it. I understood that part. Yeah.
Thank you.
I don't have any further questions. My questions were the same. Thank you.
Thank you, sir.
I just want to confirm what all samples were used to test your sensor.
So one is fluorescent dye, which is tetraphenyl TP4C, which I have labeled in the slides. Another is polyalyl amine, which is PA, that is polymer. So in the sensor, polymer and dye, fluorescent dye, they are making compound.
Sorry to interrupt. I am asking about the source of sample.
So they are all commercially.
You mentioned about like environmental, food, and human samples. So among these two, have you tested on any kind of sample?
Okay, sir. So for like we are no, sorry. I had tell about the platform for making this sensor. But for samples, we can use patient sample for alkaline phosphatase detection, and for different types of enzyme, we can use water or any other sources of that enzyme which will be present. Or like for food environmental sample, it is basis. It is depend on the which type of analyte we are using. On that basis, we can use that sample. The sensor is robust, so we can use wide variety of sample. So here we have optimized this in buffer as well as in 5% human serum.
Okay, all right. And how about sensitivity? Have you carried out any study with respect to testing sensitivity of a sensor? Yeah.
Yes, sir. So here we are using alkaline phosphatase is an analyte, and their substrate ATP. So on basis of the simple mechanism that ATP will break down by alkaline phosphatase, hence the sensitivity as well as specificity is higher. So we observed that on the solution base, we observed in buffer 0.22 milliliters per ml, and in serum 1.8 milliliters.
Can you say again your level of sensitivity, because it's not audible clearly?
Sorry, sir. So in the buffer we observed observed 0.22 milliunits per ml, and in serum 5% serum we observed 1.8 milliunits per ml.
Okay, so in your opinion, that will suffice the purpose of testing in human samples as well as other kind of samples, right?
Yes, sir. Yes.
Okay, all right. Thank you.
Thank you, sir.
Thank you, judges, for your valuable question. I now call upon code number 016 to switch on your camera. We will be presenting your video now.
Today, I will be talking about Immune Track, which is an AI-enabled IgE tracker assisted by a microneedle patch. IgE stands for immunoglobulin E, which is a type of protein primarily involved in the allergic reactions of the body. However, recent studies have shown that IgE also plays a very important role in autoimmune disorders. Autoimmune diseases or disorders are conditions wherein the body's own immune system starts attacking itself. Today's topics include the background and rationale, method, results, data analysis, and conclusions of this project. As you can see in the picture given below of the microneedle patch, it is generally considered to be more patient compliant than an injection, as it does not reach the pain receivers of the skin. Microneedles are micron-sized needles ranging from 25 to 2000 micrometer in height, and they may be made from a variety of metals, polymers, ceramic, nanocomposites, or even carbohydrates. There are different types of microneedles like solid, coated, dissolvable, hydrogel-based, or hollow. Currently, they are used in the vaccination field, obesity, for determining ECG or EEG, interpretation of the hypertension and diabetes, as well as in skin disorders. IgE antibodies are a type of antibody produced by the immune system in response to an allergen or a parasite. These antibodies play an important role in the body's immune response. However, currently IgE levels can only be detected by withdrawing the blood and having it analyzed in a pathological lab. Through this research, we aim to provide an idea into the development of a successful technology wherein constant levels of IgE can be monitored via the interstitial fluid using microneedles. IgE autoantibodies have been recognized to participate in self-inflicted damaging of the immune system responses, which leads to autoimmune disorders. Such autoimmune responses include direct damage on the tissue containing autoantigens. So, microneedles offer minimally invasive access to the interstitial fluid, which is also referred to as ISF, which is a potent alternative to blood in terms of monitoring physiological analytes including IgE. The method involves two major components: analysis of IgE levels through interstitial fluid and storage of data, as well as AI tracking, data analysis with monitoring. So microneedles consist of an array of needles which are measured in microns, and they are now currently being designed to act as biosensors by analyzing the IgE levels through the interstitial fluid. Via this project, the IgE levels will constantly be monitored, and with the help of AI, the trends can be observed, comparing it to other readings and categorizing the chances of an individual going into relapse. Relapse refers to flare up in the case of autoimmune disorders, which is very common. So AI will track and monitor the IgE levels on the basis of age, gender, type of condition, and deviations in the IgE levels. As seen through the data analysis, sometimes the IgE levels increase in the case of autoimmune disorders or decrease a lot. So the data obtained gets stored and it will be saved, directly ensuring constant track and results in the formation of graph, which will help the healthcare provider to alter the treatment plan of the patient in order to prevent further retrogression, thereby assisting in patient recovery by including applications of personalized medicine while providing constant privacy of data. A main feature of this project would be that even though all the data of all the patients will be analyzed, no leak of data will take place. Results. It was observed that the IgE levels reduce in some autoimmune disorders, or they are extremely elevated in case of other autoimmune disorders. So in the case of Hashimoto's and lupus, the IgE levels decrease a lot. And in the case of SLE, which is systemic erythromatous, lupus, and alopecia areata, the IgE levels increase a lot. Even in the case of pemphigus vulgaris, it has been observed that the IgE levels of the patient are extremely elevated. So, in UK, scientists have performed a large data set of anonymized electronic health records from 22 million individuals, which is a large-scale clinical trial, which is a large-scale clinical data analysis to investigate 19 of the most common autoimmune diseases. And in these autoimmune diseases, it was observed that a majority of women are more affected than men. Upon further observations, they also came to the conclusion that the flare-ups are very common case in all of these patients suffering from autoimmune diseases. I would like to conclude by saying that autoimmune diseases are usually characterized by flares, which also show a pattern in the IgE levels. If immune track is made possible, it will make data analysis and monitoring easy, thereby reducing delay in treatment and assuring that the conditions of flare-ups are recognized by comparing the data before they reach severity. Thank you.
Thank you, Code 016. I now invite the judges to ask their question to the respective presenters.
The presentation, I've got very, just put the answer in short so other judges can get the time. So what is the amount of work which you did practically in this till now?
I haven't done any work practically till now.
Okay, and how do you plan to use AI and microneedle together? Like you're gonna get the data and then analyze it using AI? Is that your
Yeah, the
planning?
data will be so. For example, if there are 50 patients who are using the microneedle patch, so based on age, gender, and other characteristics, the data will be analyzed and compared to a standard, and through that we will be recognizing flare-ups.
By what? You will have a AI tool you will be designing or taking by third party?
I will have to look into it. If there is a third-party tool which is available, I will prefer for the third-party tool. If it's not, then it needs development.
Okay, well, you can't use third party tool because of now government of India has made it mandatory to have a ethical legal requirement for all the patient data, which cannot be used for third party. We will have to think about how you analyze the data. Yeah, that's all. Only two questions from my side. Thank you.
Thank you.
Sorry, which data set are you going to use to train your models, and what model are you going to use?
So we will be once the project is ready, we will be using we will be actually asking people who are suffering from autoimmune disorders to like for we will be asking people suffering from autoimmune disorders to kind of just wear the patch, and through that we will be collecting the data and then making a standard of it.
Okay.
So it would be microneedle based wearable patch, right? You are proposing.
Yeah.
Yeah. Any thought about on the material or design aspects with respect to the microneedle?
I haven't really thought about it till now.
Okay. All right. Thank you.
Thank you.
Thank you, judges, for a valuable question. I now call upon code 017 to switch on your camera. We will be presenting your video now.
Respected judges, teachers, and my dear friends, our project titled Predict RX Care, an AI powered personalized food drug interaction and medication management system. The core idea is simple to make the medication therapy safer and smarter using real time analysis instead of basic reminders. Presentation overview. So, in this presentation, we will look upon the background problems, limitations of existing apps, why predict RX care, how it works, and many more. Introduction. Now, let us start with the background first. Medication errors are a major global healthcare challenge. Every year, millions of patients or people are affected due to the wrong doses, missed doses, or timing errors. So, food drug interactions are often overloaded or overlooked or ignored, which ultimately reduces the efficacy or results into serious side effects. Similar looking pills and polypharmacy increases the complexity and especially in the elderly patients. Existing app focus only on reminders. Like instead of simply reminding someone it's 8 a.m., take your pill, Predict RX Care can say, wait, you just ate breakfast 10 minutes ago. This, this medicine must be taken on an empty stomach. I will remind you again in 30 minutes. This demonstrates the difference between a typical reminder app and an intelligent system. Next slide, please. Medication errors, incorrect timing with meals, missed or delayed doses, confusion between similar looking pills, lack of food, drug and drug checks, polypharmacy. These are all the predictable medication errors which arises using the existing apps. So what are the existing apps? We will focus on the next slide. Next slide, please. Existing solutions. So there are many popular apps like MediSafe, MyTherapy, DoseCast, and what they provide is basic dose reminders, dose history tracking, limited adherence notifications. But there are severe limitations that there is no any data regarding food drug or drug interaction analysis, or there is no dose adjustment based on meals or missed doses, or there are no personalized risk scoring factors or assessment factors. Why Predict RX Care? So Predict RX Care is the solution of the limitations of the existing apps. It is the personalized AI driving dosing adjust to user routine and meals. It detects the high risk interaction before the harm occurs to a patient. Also, there are many pill verification tests which is done using camera which ensures the correct matches of the medicine. Also, mail-linked reminders are there which reduces the missed or ineffective doses of the medication. Also, multi-mode accessibility like audio, vibration, or visual alerts, which will benefit all category type of patients. Also, to put it simply, imagine a world where your medication app thinks like your personal pharmacist. It knows what you ate. It knows which pills looks like. It knows which drugs interact. It adapts to your schedule. It keeps you safe every single dose. And this is what Predict RX can bring. Now, I will request my colleague to please continue with the topic. Thank you.
Thank you. Now I will explain how the Predict RX Care actually works. The process starts with the pill scanning. The user simply see the medicine using the camera and system matches it, verified with the standardized drug data sets. Next, the user enters the meal details using the nutrition, food composition data along with the meal timing and the daily routines. The personal factors like age, habits, allergies are also considered. This combined input goes into the AI engine. The first layer uses the rule-based logic for the immediate checks like food-food interaction and food-drug interaction and drug-drug interaction. The second layer uses the machine learning models to detect the patterns in how the users take the medicine and how the meal routines affects the drug action. Based on this, the system dynamically adjusts the dose, safe dose timing, and the system generates the alerts like meal-linked reminders, dose adjustment, high risk warning. Finally, everything appears on the dashboard where the user can get the clear view of the medication schedule, risk, personalized recommendation. Now, we will discuss the clinical impact on the users. To understand the impact, we evaluate how the user behavior changes before and after the Predict RX Care. The medication adherence improved from the 55 to 87%. This happened because the user received the dose reminder where it aligns with the meal timing and the interaction risk. We create a risk index score which shows the score from 0 to 10. 0 to 3 are safe, 4 to 7 is indicate the caution, and 8 to 10 requires the high and immediate attention. Our measurable outcome were strong, like 32% improvement in one-timing dosing, 40% reduction in food-drug interaction errors, and 78% of users interacted actively with the system, meaning they understood the therapy better. Overall, it duplicates the dosage drop. Colorblind users can get the clear alerts and the physician receive the better real-time patient dashboards. Next, we will see the inclusive design for all the users. Medication app usually assume every user is the same. We don't make the same mistake. Our interference is designed to be inclusive for the all type of user. For the colorblind users, we use the dual color palette system. Visually impaired users can get the voice-enabled navigation and text-to-speech instruction. The deaf people can get the hepatic vibration alerts, and elderly can get the large text UI. Low literacy users can get the pictorial-based instruction, and even illiterate can use the camera guide pill identification. Every design is tied up to the specific beneficiary group, and hence we can conclude our system can be useful for the all people. Further will be explained by my colleague. Thank you.
This slide shows the future growth and expansion opportunities of our system, which are as follow. First is predictive risk forecasting, which uses long-term user data to predict future interactions. For example, predicting likely food drug interactions and risk factors. Another is integration with lab reports. Our system automatically reads and interprets lab reports such as liver test function, renal function test, and lipid profile or glucose levels. Then next is clinical dashboard. Doctors get real-time monitoring of patients, which enables food drug safety and risk factors. Another is smart refill and follow-up system. The consumer or user get reminders when the medicine stock is low, which ensures continuous medicine without any gap. Then another is multi-platform system. Our app can be used by Android, iOS, and web dashboard system. Expanded database and global compatibility by making changes in region, food interaction system, and multi-language support. Our app can be used in various regions and cultures. Next slide is conclusion. Predict RX demonstrates how AI can enhance prediction accuracy by making changes. It also helps clinicians make better and faster decisions. It provides growth, and by ethical support, by providing ethical support, it can address a large-scale adoption. Thank you.
Thank you, Code 017. I now invite the judges to ask their question to the respective presenters.
Yeah, a good presentation. I just wanted to know, is this just a concept or is your app ready?
No, we have this is just predictive analysis of our model. Like we have predicted many key outcomes by the limitations of the existing apps. We have worked onto it and we have made something improvement which can be comes into idea later.
So this is just an idea, right? Then how did you come across that outcome 55 to 87% outcome improvement it happened? How can you say that if it is just an idea?
Sir, it is when we have predicted that key outcomes like medication adherence improvement, which is rises before Predict Rx care, it was 55%, and after Predict Rx care, it was 87%. It
How
was
did you
just
make
taken
that?
as an average.
How did you
Okay,
make
okay.
that?
We have actually we have using the formula of adherence improvement, that is correct dose taken divide by total dose taken multiply by 100, and according to it we have taken the average of it, and we have predictively 87 is the average of 84 to 89%. 89% it lies, so we have taken just average.
Okay, thank you.
And
Thank you.
these are
Yeah, thank you. Thank you. I understood. Other judges can take over.
Thank you. I had the same question what Anup sir had, so same for me also.
Thank you, sir.
Yeah, I just have one or two questions. So end user of your system would be patient as well as physicians, right?
Yeah, because doctors and pharmacists all wants transparency. So our app will give transparency. How? Because our app itself suggests like predict RX care. RX means something related to prescriptions. When doctor provide the prescription and pharmacist dispense the medicine, the correct intake of the medicine by the elderly patients. Not only our app gives the prediction, but also it gives the reason behind it. Like that.
Okay. What is minimum requirement of your system, like in order to use it? Just a mobile and internet and an app. Correct, right?
Okay. So in our system, first of all, when every user login, it does not make any region difference. Like we can select any local languages also in that. And when the user inputs some data or a manual, means full medical history or user which have to fill that data, then fill verification steps after using a camera. Also, AI engine is designed which have many validated sources like WHO, US FDA, then drug bank to compare the drug and drug food interactions, and similarly predict the risk assessment interaction factors. And based on it, every user, like if colorblind user is there, then we use large icons like danger sign, vagera, and then the person gets a proper risk assessment factor that his particular drug lies on which category, like safe, caution, or high risk category. Like for example, if a person is taking beta blocker drug like atenolol, when it is consumed with the high fat diet, it will decreases the drug absorption and ultimately reduce the efficacy of the drug. Similarly, you can take example of NSAIDs like ibuprofen, which should be taken on the empty stomach, or otherwise it can cause irritation into the GIT tract. So our app focuses on that. Similarly, also we can take example of vitamin D, which when taken with an antacid, it will reduces the drug efficacy. So therefore, existing apps won't predict this, but our app like Predict RX Care will immediately flag that this combination is a little warning type. You take vitamin D 2 hours ago. Why? So that all predictions like decreases the drug absorption rate and all vagera. This
Okay.
app will not
Yeah.
only predict but
But
explain
with
it further.
respect to this, in order to do all this, in case of especially the illiterate people or the low literacy people, some assistance would be required that is desirable, right? In order to do all this and achieve all this.
Yes,
Am I right?
for clinically. Yeah, yes, you are right.
Yeah. And one feature you mentioned that smart refill and follow. So how does it
Yeah,
work? Like, do we have to enter the
sir, it
data, for
was the
example, if I
future
purchase?
opportunities that future. Yeah, sir. It was the future opportunities like on upgrading of our app, we can use smart refill system. Like we can book the medicine from app itself, but it is not learned. Now we are focusing upon it is the future opportunities or the work we can do later.
Okay, all right. Thank you.
Thank you,
Thank
sir.
you, judges, for your valuable questions. I would like to now call for 022 to switch on their camera. We'll be presenting your video now. Code 022, can you please confirm you are there with us in the meeting? She's in a meeting.
Yes, ma'am. Yes, ma'am.
Okay.
Hello, everyone. We are presenting a project titled AI-assisted multi-biomarker liquid biopsy interpretation system for early cancer detection. Our project proposes a novel way to detect early-stage cancer using blood-based biomarkers interpreted through artificial intelligence. We are Team AI-022. In many cases, early stage cancer doesn't show clear symptoms. As a result, patients are often diagnosed late. When treatment becomes harder and outcomes are poorer, traditional detection tools like CT scans and tissue biopsies are either invasive or limited in sensitivity, especially in early disease. Liquid biopsy solves part of this problem. It uses a simple blood test to detect tumor derived molecules like DNA, RNA, or proteins. But the limitation is that most current tests rely on just one biomarker, and this often isn't enough. Our idea is to combine multiple weak biomarker signals using AI to detect faint cancer signals earlier and with higher confidence.
Our aim is to develop a system that uses artificial intelligence to analyze three types of biomarkers: ctDNA, exosomal miRNA, and circulating protein, all extracted from a blood sample. The system will be developed using Python for the AI model, SQL for data handling, and CSS for creating a user-friendly interface for doctors. The final user will be a clinician, not a patient. The model will output a cancer risk score that helps healthcare professionals identify high-risk cases faster.
Our system integrates three classes of biomarkers: ctDNA, which are short fragments of tumor DNA that circulates in blood. Cancer cells shed this DNA as they die. Its irregular pattern can indicate tumor presence. Second, exosomal miRNA. These are small RNA molecules packed in vesicles called exosomes. They are involved in gene regulation and are often altered in cancer. Third, circulating proteins. These include known tumor-related proteins like PSA or CA125. They are not always specific, but they add useful physiological context. By combining all three, we aim to build a much stronger detection signal than using any one alone.
The prototype workflow is designed around three tools: Python, SQL, CSS. Python will be used to build and train the AI model. SQL will handle patient data queries and storage. CSS will style the interface used by clinicians. Once biomarker values are fed into the system, the AI model processes the inputs and outputs a cancer risk score between 0 and 1. This score summarizes the probability of cancer based on all biomarker signals combined.
Our methodology begins with collecting archived plasma samples from diagnostic labs. This includes both confirmed cancer cases and healthy controls, matched for age and sex. Clinical metadata like cancer stage or treatment status is also considered. Blood is drawn using standard protocols and processed quickly, ideally within 2 hours, to isolate plasma. The samples are then stored at minus 80 degrees Celsius to preserve biomarker integrity. From these plasma samples, we extract 3 different biomarker types. ctDNA is extracted using silica column or magnetic bead kits. This includes both healthy and tumor-derived DNA fragments. Exosomes are isolated via ultracentrifugation or kits. RNA is extracted from these vesicles to study miRNAs. Circulating proteins are collected from plasma or EV lysates, preserved for immunoassay like ELISA or mass spectrometry. After isolation, each biomarker undergoes characterization. For ctDNA, we use shallow whole genome sequencing to analyze patterns like fragment size, end motifs, and genome coverage. These fragmentomics feature reflect tumor activity. For exosomal miRNAs, we use either RT-qPCR or small RNA sequencing to detect unknown cancer-related miRNAs. For proteins, we focus on tumor markers and inflammatory proteins using immunoassay to quantify them.
Methodology, data processing, AI fusion, and risk score. Once we collect all the raw biomarker data, we start data pre-processing. Because DNA, RNA, and protein data are on different scales, we normalize each data set, such as reads per million for ctDNA or concentration per microliter for proteins. We remove low-quality samples and then extract key features. First, from ctDNA, average fragment size, short fragment ratio, end site motifs, and chromatin coverage bias. From miRNA, normalized expression levels or ratios of specific oncogenic versus suppressor miRNAs. From proteins, absolute or relative concentrations and expression patterns across groups. Since this creates high-dimensional data, we apply dimensionality reduction like PCA or use feature selection techniques. Example, LASSO, which stands for Least Absolute Shrinkage and Selection Operator, to filter out noise and retain the most informative markers. Next, we build the AI model. We use boosting algorithms such as XGBoost or LightGBM to combine features from all the 3 biomarker types. Models are trained on a split data set. Example, 60% training, 20% validation, 20% test, and validated using cross-validation. To enhance performance, we can use an ensemble model where separate submodels are trained on DNA, RNA, and protein features, and their outputs are combined via meta-model. The result is a unified, interpretable cancer risk score scaled from 0 to 1, representing the system's confidence that the input biomarkers suggest cancer. This score helps clinicians prioritize follow-up investigations.
Our data analysis focuses on how well the fused AI model performs compared to each biomarker used individually. We now have validated performance estimates from literature. ctDNA alone shows 70% sensitivity in early cancer detection. Exosomal miRNA achieves 75 to 80% accuracy depending on cancer type. Circulating proteins like CA125 or PSA have moderate accuracy, typically around 55 to 60%. However, when we combine all these biomarkers using AI, previous studies report accuracy improvements to over 90%. For example, a multi-analyte test by Liu et al. Integrating cfDNA methylation and proteins achieved up to 98% sensitivity and specificity for multiple cancers. In our system, the AI model fuses weak but complementary signals. We simulate classification into low, moderate, or high risk categories based on the cancer risk score between 0 and 1. The goal is to support the doctors in flagging high risk cases early, even when no clear symptoms are visible, and reduce missed diagnosis.
In conclusion, our system shows that when biology is combined with AI, we can overcome limitations of traditional testing. Liquid biopsy is already a promising technology, and with multi-biomarker fusion, it becomes even more powerful. Designed for clinical use, this tool could guide earlier diagnosis and potentially help save lives by reducing missed cases and diagnostic delays.
These references support our biomarker selection, AI integration strategy, and system design. They are based on peer-reviewed studies in cancer diagnostics, liquid biopsy, and multi-analyte testing. Thank you.
Thank you.
Thank you, Code 022. I now invite the judges to ask their question to the respective presenter.
Thank you for the presentation. I have a few questions. Like, first of all, how do you make sure that your product is better than what is already there in market? The second question is like, how effective it will be to detect the early, like when you say detection of cancer early, how early can you detect that thing? Can you detect the pro-biomarkers for this based on the other initial markers which is there in blood before actually the cancer arrives? Can you also predict the migration of the tumors which might be happening? There are two questions from my side.
Yes, sir. So basically, the previous studies focus on signals of a single biomarker, due to which during early stage of cancer, the biomarker signals are very weak, which leads to missed diagnosis, which is why we focus on combining mainly three biomarker signals together, due to which we presented the fusion method, uh, uh, which will give us a better signal, such as strong signal, and then lead to cancer risk score, which is the evaluation parameter for the detection.
Sir, we can't hear you.
Can't hear you.
Okay. How do you make sure that there is no false positive sign due to inflammatory reaction which does happen sometimes with the biomarker?
Wait, sir, can you repeat the question? Sorry.
How can you make sure that there is no false positive due to biomarkers? Sometimes there is inflammatory gene, and sometimes there are some mutations which can cause those particular biomarkers to be in higher level, which is in cancer. How do you minimize that part?
Sir, by using instead of to avoid that confusion, we are fusing three different types of biomarker and then integrating it into the system, and which gives us the output of a cancer risk score.
So firstly, there will be a step. Firstly,
Continue.
when the so when the data is inserted into the model, firstly there will be a single biomarker separately analyzed first, and then all the three biomarkers will be clubbed, such as fused together, and then we'll evaluate it. So due to which, even if there is false negative in one signal, but due to the analysis of combined three signals, we can detect the cancer more accurately.
Okay. Thank you.
Yeah, my question is, suppose if you get blood today from a patient, okay, how long does it take for you to arrive at your final conclusion?
Once there was we mentioned it in the methodology part, which is first one, sample collection and processing. We mentioned that once the plasma is collected, it will be stored at -80°C for approximately 3 hours, and then it can be analyzed better.
No, my question is, how long would it take for you from the sample collection to your final report?
Final evaluation report, right?
Like from blood
Yes.
test.
Yeah, so it can take approximately 1 to 2 days. Yeah.
1 to 2 days. Okay.
Okay, so you have developed prototype of this study, right? As mentioned in the presentation.
Sorry, sir.
Sorry, sir.
You have developed prototype.
No, sir, it's a conceptual note.
Okay, fine. So questions are already covered by previous judges, so no question from my side. Thank you.
Okay,
Thank you, sir.
Thank you, judges, for valuable questions. I now call upon code number 026 to switch on your camera. We'll be presenting your video now.
Good morning, respected judges. Today I am presenting abstract code AI 026 AI-driven predictive stability modeling for solid oral dosage forms. So these ones explore simple idea. Can you understand how tablet will degrade? How long before waiting months for real-time stability? So background and rationale. So in oral solids, stability is not just a test; it's major bottleneck. Single real-time test takes 6 to 12 months, and in India, 40 degrees Celsius to 75% RH degradation accelerates dramatically. So the impact is huge. Around 20 to 25% of batches end up repeating stability. Each failed cycle can cost 20 to 30 lakhs just months of delay. And the root problem is straightforward. We only understand degradation after the study finishes. By that time, batch is already dead, lost. So the question becomes, what if we could see these trends early? What if formulators knew upfront how packaging, excipients, and climate might influence degradation? So here an AI supported early prediction frameworks become valuable. So now next slide. So methods. So I have written these methods in this slide. So method, data and features. So to build this idea, we map the most influential features. From APIs, we consider log pK, melting point, hygroscopicity, and even oxidation tendency. From packaging, we include barrier strength and water vapor transmission rate, and from environment, we use temperature, humidity, ICH zones. Simple descriptor tools like RDKID and published SPL data help structure these parameters. Together, they create a picture of how a formulation might behave over time. So, next is AI modeling pipeline. So, once we define that data, modeling workflow becomes the quite intuitive. So, we begin by cleaning and normalizing inputs. Then we engineer features to amplify meaningful patterns. A hybrid model works well here. A random forest layer highlighting which features dominate, and an ANN layer captures the nonlinear behavior seen in the real degradation. From here, we generate simulated degradation curves and categorize formulation into low, moderate, or high risk. It's a workflow that balances simplicity, interpretability, and usefulness. So our objectives. So I have divided this method slide into objectives. So our objective is to propose a concept level model that supports early decisions. We aim to estimate potency, loss curve, predict impurity growth, and like high risk formulations before they enter long-term studies. We targeted stimulated performance with R2 that is less than or equal to 0.8, enough to guide development, not replace regulations but to provide early visibility. Simulated results. This graph represents a simulated trend based on literature, a way to visualize how the model would behave. At 25 degrees Celsius or 60% RH, potency stays close to 93.4 at month 6, with only mild decline. At 40 degrees Celsius or 75% RH, potency falls faster, reaching around 81.9% at month 6. The variation in this prediction stays within plus minus 2.4 to 2.9 simulation work. So here is the key finding of the results that I have explained just now. So what this shows is simple, what a conceptual model can anticipate the shape of degradation much earlier in development. So a clear separation between stable and unstable profiles observed. Data analysis. When we interpret these patterns, some signals stand out. Humidity emerges as the strongest driver. Temperature and excipient interactions shift the curve further. APIs with high hygroscopicity decline early, especially under tropical conditions. Packaging with low barrier scores tend to fail sooner, and the clustering helps to separate stable and unstable APIs. So the main takeaway in this. Such insights could guide teams towards better packaging and smart excipient choices and fewer conclusion. To conclude, the predictive stability concept aligned with the direction of Industry 4.0 is pharmaceutical development. If implemented, it will save 4 to 6 months per formulation, reduce 30 to 40% repeat stability runs, and lower the financial losses caused by the late stage failures, especially in India's climate. It deserves consideration because it targets real high impact problems. It is a practical approach and use formulation teams early intelligence without interfering with the regulatory requirements. So, before I close, I want to highlight one important point about why this concept matters right now. So, the pharmaceutical industry is moving towards faster, data-supported development, but stability remains stepping slow. Traditional cycle, every decision is formulation from polymer, concentration to packaging material. Eventually, waits for stability data, and whenever the batch fails, entire project reset. It is not just academic; it's directly affect development and timelines, cost to companies, and patient access. Why my concern? The policy is not a replacement for ICH guidelines, but a pre-stability intelligence layer, a tool that helps teams make smarter choices even before the first long-term chamber run is completed. Formulators can see early signals such as high hygroscopy APIs or moisture barrier packagings or excipient incompatibilities. They can act earlier, reduce trial and error, ultimately deliver a more robust product in stability cycle. So the value of this idea is very simple. We are shifting stability from a reactive process to a proactive one, and even conceptual AI-assisted prediction has the potential to save companies months of time, prevent costly redesigns, and support faster data-driven decision making. This is the core strength of this proposal. Practical, realistic, and strongly aligned with the India's formulation challenges. So here I finish my presentation. Thank you. I am ready for the question answer session.
Thank you, Code 026. I now invite the judges to ask their question to the respective presenters.
Thank you for your presentation. So I have one question. When you say degradation, with respect to what are you predicting degradation?
Degradations. Okay. So degradation about API, the other properties, excipients.
Excipient won't degrade. It comes with API. So the main, when you go to industrial point of view, we have nitrosamine, which is very important critical attribute.
Sorry, sir.
So the main attribute which industry looks now is nitrosamine. The main degradation which happens nowadays. Even FDA has got a guideline for that thing. Will you be able to predict the nitrosamine content of tablet using this technique?
Yes, sir, it can be done because, sir, as I have explained that it works on what data you have given to the model. So if, as it is new formulation, it might be some limitations, but as however the data we provide to the model, more and more data will provide, it will develop and it will give a 100% real predictions as the time passes.
Evolving model, you mean to say it's not a constant, it's evolving. So it might not give the data in the first place. Maybe you use it for 10 times, then you give more predictive data. That's right?
Yes, sir. Means how many times we will use it, it will become more and more better as more data we will provide to the model. So it works on that.
So it's not plug and play model. We have to train it. We need to put multiple tasks, multiple modeling studies. We have to keep a lot of data. I mean, we have to do a lot of studies before we start to use this study. It's not pre-trained machine learning program.
Yeah, so it will use all that stability data, all the features and what the data the industry or the company have. So it will be trained by the company first, but afterwards it will work specially on the basis of information.
But the one industry data, they won't let it used by other industry. So every industry has to do the same thing from scratch. If I am an industry, I won't give my data to you to sell it to some other company. What will you do in that case?
So this model will be used at basic level, means one industry will use at this model, and it will work differently, sir, according to their conditions.
Okay. To you, Anup, sir. Thank you.
Have you done any predictive modeling?
No, sir, not. It
You
is
have
only
not done.
conceptual.
No, not this. Any other type of predictive modeling have you done?
No, sir.
That's all from my side.
No questions from my side. You can go ahead.
Okay, thank you.
Thank you, judges, for your valuable questions. Now, we request the judges to share a few words about their experience on today's event.
Yeah.
Yeah.
It was a
Anup,
very
sir.
interesting. Oh, sir, please carry on.
No, sir. I was inviting you to share your thoughts. Yeah, please.
Yeah, it was a very interesting session. So we found a lot of interesting ideas, and some of them are very relevant to the industry. Especially the last one, at least two or three we found very relevant to the industry. Then there are patient focused, and then there are physician centered. So many different types of ideas students have performed, and they have done a basic, quite a basic reading. I would suggest that people run a bit of modeling, and I understand how, and I urge them to understand how complicated certain situations get, so that it would be easier for them to focus on any particular area, so that they can refine their ideas. Some of the ideas were very refined, very happy to say that. A few of them were very broad. So they can improve upon that once they learn more things about this, they will be able to. But I'm very happy that it's not the idea that matters sometimes, it is the way of thinking of the students that is more important. So I congratulate everyone for your innovative ideas.
Thank you, Anup, sir. Just one question. All of them are PhD student or are they master's student too in the group?
No, so Dr. Abhijit, if I may interfere here. So I just wanted to share with both of you that we have entries from even UG, PG, PhD, not only pharma. We have entries. So for AI in healthcare, we have not kept. In Hackathon, we have not kept anything like the only PhD or only PG. Anybody of life science sector, healthcare sector, they can submit. So we have received entry from across India, all 28 states and 4 union territories, of course, in all 5 categories. So that was the involvement. We have around 230 institutes have taken part into all these 5 categories. So probably Afrin madam would be able to tell us that in AI in healthcare, which all states, if you don't reveal the name of the institute, but which all states have taken part and assigned entry in AI? Dr. Afrin, if you can highlight.
Yes, ma'am. Ma'am, we've got entries from across. So in AI, we've got Maharashtra, we've got South as well, and even from 2 entries from even Assam.
Okay.
Okay. So I would say that the type of work done is really nice. If I compare with what we were when we were in bachelors and masters, these things were like for us only for industry. We never thought to touch these topics because it was way too high from what we were taught at that time. And I mean, obviously, the things have changed a lot, and the way they have been working, these topics, most of them, they are mostly topic on PhD, if we think from my point of view. So it's really up-to-date topics, and few topics like lab on chip for using the phosphatase on paper-based chips. So these are really cost-effective, commercializable idea. and not just a dead research what we used to do in past. Something which is tangible outcome is there. We have product from the research what we've done. So that way is a really good thing. And I guess not a lot of people have been attending few more AI hackathon, and they don't have a lot of product based presentation, which was there in this research congress which I saw. Students are with the product what they want to make. So they are very clear on the outcome of what they want to do. And that's a good thing, in fact. I mean, the way academic is taking on this thing to make it more product tangible outcome and not just keeping it as something which is in conference, just presentation, put some theoretical research. So it's a really something, and we got to know a lot of new things. I mean, now we feel we need to read more to compete with what our students are doing. So that's a good thing for us also. We have to update ourselves to make sure we are in line with what people are doing right now. So thanks for giving us a chance to actually be a part of this presentation.
That's wonderful. Thank you very much, Dr. Abhijit and the team. We, in fact, request you to stay back for valedictory, which we would start once all parallel session gets over. You will be sharing the results by putting your signature and send the document soft copy in email itself. The email from which you received the scorecard. So you can send that. If you need to talk to each other, I would request Afrin madam to have a breakout room if they want to discuss, three of them, and they can come to the final decision.
Okay, ma'am, I'll do that now.
Right, so it would be easier for three of them only to talk and then come to final decision. But we would request three of you to remain present during valedictory, because we would be more than happy to join the ACG team in declaring the results. And then only we would come to know whether the student was PhD, PG or UG, because at present we would not know because it was completely anonymous. So we would ask student to talk about themselves when we declare the result. All right, over to you, Afrin madam.
Yes, ma'am. Dear judges, we've sent you the score sheets. I'll just create a breakout room for all three of you and post it in the chat box here.
A special note of appreciation goes out to all the participants
in the attendees list? Namira and Isha. Technical team, can you quickly check if there is the number? Probably they must be as per the number.
Yes, ma'am.
They must be PG 98 and PG 41. Do we have them? All right.
Okay, let's move on.
Let's move on.
Our
Hello.
next.
Yes, there is.
Oh, do we have someone?
Who is this?
Ma'am, just give us a minute. We are adding the
ma'am.
audio.
98.
Say it again.
PG 98.
No, the winner. Yes, Namira.
Yes, ma'am.
Yeah, you can say something. All right, there is some issue. Should we go ahead?
Yeah, the next category we would like to announce the awards is like for the very first time we had the hackathon oral solids. Sorry to interrupt. Ma'am, we are sorry to interrupt. We're just adding the audio, the clapping audio. Just give us 2 minutes, ma'am.
So then, meanwhile, we want someone to speak.
Behavior.
Namira, are you saying something? All right, so there is some issue. So meanwhile, probably I'll be just addressing everybody. So thank you. The huge thanks to the entire student team who is working behind. Okay, I'm unmuting myself. Sorry, I see that I was muted. So meanwhile, let me thank my entire student team who is behind this entire online activity, managing every activity. So we have the activity going on 3 different rooms, and we have one group which is in. Can somebody mute? I want one student to be constantly watching this and immediately muting the person who is in audience. So we thank the student entire community and the entire group who would be coming at 8:30 or 9 o'clock and making sure that everything is in line. All the slides decked up properly and the smooth coordination between all the parallel sessions. So thank you, students, community, entire for your wonderful, and I'm sure it was a learning for you. Managing all this online event, it's learning. It's much more learning than your books, what you prepare for exam. So thank you for coming forward to manage this entire show. And congratulations to all the winners. Over to you now, Parizad madam and Subhashis sir and Dhanraj sir. Now I think we are ready with announcing the winners of PhD. How do we go? Next is oral solid and PhD last.
Yes, next is hackathon oral solids.
Go ahead.
Unfortunately, we do not have any third prize being announced. We just have two. The second position goes to OS 019 Sayali Hingane from Singer Technical Education Society, Srimati Kashibai Nawale College of Pharmacy, once again Pune.
Congratulations. So we have a person here. So you can put your camera on, and if you would like to speak once we declare about both the winners.
Right. And the first position goes to OS 004, Chetan Rajote, YB Chavan College of Pharmacy, Sambhajinagar. Is Chetan around?
Can we have the
Hello, ma'am.
certificate of Chetan?
Yeah, here is continuous next. Thank you so much.
Yes, Chetan, can we have your camera on?
Okay.
And if you'd like to say something.
Hello. First of all, thank you so much for organizing this type of hackathon competition for oral solids. I really enjoyed the competition, and thank you for giving me chance to present my innovation in your competition. So thank you so much, and thank you so much for the first prize. Thank you so much.
Are you the PG, PhD or UG student?
I am post-graduation student, PG student, Master of Pharmaceutics.
Wonderful. All right. Thank you. Congratulations.
Thank you so much. Thank you. Thank you, ma'am. Thank you.
Thank you. So wishing all the winners all the very best from our side. We can go to the next one, Subhashish, Hackathon Artificial Intelligence.
Yeah, I'm waiting for the declaration on the screen.
Yes.
Okay. So now comes the AI hackathon. The second runner-up is AI 009. Shraddha Sonavane and Homi Bhabha National Institute, Mumbai.
Congratulations.
The first runner-up is AI 022. Nidhi Ajmera, sorry. Again from BNCP, Mumbai. And here comes the first prize, which is AI 002, Sanket Palve from DY Patil College of Pharmacy, Apurdi, Pune. Congratulations to all three. Great job done.
Thank you.
So during the competition, the judges wanted to know whether they are from UG, PG or PhD. So we can request the winners, Sanket first, if he is there. And then we can have Nidhi and Shraddha, if they are there, they can tell us, are they from PG, UG or PhD? Sanket is there?
Yes, ma'am, I'm here. Good afternoon. I'm Sanket Palve and I'm a student of PharmD, Doctor of Pharmacy from Dr. D.Y. Patil College of Pharmacy, Kurdi, Pune. So I'm in my PG itself because I'm in the 6th year.
Wonderful. Congratulations.
Thank you.
Do we have Nidhi?
Yes, ma'am. Hello.
Tell us about you.
Yes, so I'm a student. I'm an undergraduate student from BNCP.
Wonderful. Congratulations. Do we have the third number?
Thank you, ma'am.
Yes, ma'am. I am a second-year PhD student.
Wonderful. Congratulations. So I had requested judges of AI Hackathon to remain present if they are. I mean, they would know that they are the PG UG students together. Over to you.
So, good afternoon once again. I hope I'm audible.
Yes, sir. You are.
Yes.
So, I'm here to announce for the PhD category oral presentation. I'll start from third, as usual. The second runner-up goes to PhD 011, Rian Verghese from Philadelphia College of Pharmacy.
Congratulations, Ryan.
Congratulations.
I know it's early morning probably there. If you are there, you can talk once we declare all three.
Now coming to the first runner-up, it is PhD 012 Smriti Bhardwaj from Naipur SAS Nagar.
Congratulations, Smriti.
Congratulations, Smriti. Now the first prize goes to Sayali Dige from Naipur, Sasnagar again. Congratulations. Congratulations, Sayali.
Congratulations, Saili. So do we have the winners here available?
Hello.
Yes. You can put your
Ma'am Saili.
camera on
Yes,
and tell us
my
who are
camera
you.
is
Yeah.
on.
Okay.
I'm Saili Dige from Naipur, Mohali, and I would like to thank everybody. Special thank to Afreen ma'am for coordinating the event so smoothly. Apart from that, thank you to Dhanraj sir and Gaikwad sir for all the suggestions, for all the insights. Also, Vasanti ma'am. And last but not the least, thank you so much, Munira ma'am, for this platform. I have been participating in SRC since I think in my master's, this started first Student Research Congress, and I will continue to participate. Thank you so much for the platform. Thank you.
Wonderful. Congratulations. Congratulations.
Thank you so much. Thank you. Thank you.
Do we have any other members?
Hi everyone, I'm Ryan.
Winner? Yeah, hi Ryan.
Hi, Dr. Momin. Thank you so much.
Congratulations.
Thank you so much. I really appreciate it. And well, I was actually flying back to India for the winter break. So I'm actually connecting from India today. So
Okay.
yeah,
Great.
and I really appreciate the team for conducting everything super smoothly, and they've been really supportive over the entire process. Since I was traveling, I told them that I may not be able to make it, or they've been really very accommodative about the entire process. So I really am very appreciative of it, and the topics were indeed really good, like something that we actually need at this point, like be it
Wonderful.
innovations.
We look
Of
forward to
course.
your school to participate every year.
Absolutely, we will. We certainly will. And in fact, I'm also partially representing my alma mater, Pune College of Pharmacy.
Wonderful. Okay, good. Congratulations.
Great. Thank you.
Congratulations. Yeah.
Thank you so much.
Congratulations. Yeah. Yes. Yes, over to you, team.
Your achievements are a source of immense pride for all of us, and we eagerly look forward to seeing the lasting impact of your work on the field of healthcare. Keep pushing the boundaries of research and continue to shape the future of healthcare with your passion and dedication.
As we conclude this remarkable event, let us carry forward the spirit of innovation and collaboration that has defined the Student Research Congress. The theme, Innovations and AI in Healthcare, is not just a tagline, but a call to action for all of us to strive for excellence and contribute meaningfully to the society. We hope the Student Research Congress has inspired new ideas, forged valuable connections, and sparked a renewed passion for research that will drive transformative advancements in healthcare.
Once again, I extend our deepest gratitude to all our guests, sponsors, judges, faculty members, students, and volunteers who have made this event a grand success. Your support and dedication have been invaluable in bringing this Student Research Congress to life. Let us take a moment to celebrate our collective achievements and look forward to an even brighter future, filled with more groundbreaking research, innovation, and collaboration. Together, we are paving the way for a healthier tomorrow. Thank you once again, and here's to a continued success.
Can we have everyone to put their cameras on so we can take our own selfie here? So I request everyone to just put the cameras on, and the entire team who is behind this entire activity, if you can turn your computers.
So everyone is
and
getting
then we
ready. You
have
see now?
everyone
They're getting
together.
ready.
Do we have other cameras? Do we have Lokesh sir and all other faculty, Siddhi madam, those who have worked together, Akshata, I see Chintan sir, and the other group of students, the technical group who is on in the other room. And let's request everyone to keep their cameras on so we can have a large gallery. And we can have a picture together. So I request students to take screenshots. Afrin madam, your screen is gone. We don't see anything. That is the main computer from where the entire event was operated. We see completely white background of it. Yeah, now we see. Now it goes. Yeah, there you go. The entire team. All right. Thank you, everyone. Appreciate your wonderful coordination. And thank you. I'm sure it was a learning experience. And thank you all judges and our guest. So selfie, honey. You see, they are taking selfie there. I said screenshot is a selfie. Are you taking selfie there, guys? All right. Thank you, everyone. Thank you, Dhanraj sir. Thank you, Subhashish sir.
Bye-bye.
Thank you, Dr. Parizad. Thank you very much. And thank you all the participants and the winners. Congratulations, everyone. All my anchors and student coordinators. I see all of you. Thank you very much. Thank you. All right. You're muted, Parizad. You're on mute.
Yeah. Just wanted to thank complete BNCP team and Dr. Munira yourself for your leadership, the way you have taken this forward. It's really excellent. Thank you.
Thank you. Thank you very much. Yeah. Thank you.
Look forward
everyone.
to next year. Bye.
Yes. Look forward to see you next year, all of you. Thank you. Bye.
Bye.
लोकेश सर बहुत सेल्फी हो गई. Look at all of you. Thank you, everyone. Guys, you did wonderful, I must say. You did wonderful, wonderfully coordinated. The only problem is मेरी claps का आवाज नहीं आया. No problem. But you did all awesome. So 5-star to all of you and the kudos to all of you. Thank you very much. Thanks. Yeah, there you go. Now I see the sound coming. All right. Take care, all of you. Thank you. You always make us proud. Proud of you all. First year, second year, third year, final year, MFARM, everyone, you make us proud. Keep growing, keep learning, and wish you wonderful, wonderful professional life ahead. I'm sure this all coordination will help you a lot further in your research, in your studies, and in your professional career. So thank you, all of you. Bye. See you.